Thank you. Sarah already introduced me, but my name is Jerry Sharpe. I'm from Telluride High School, and this summer I studied computational biology at the Neretti Lab at the Brown University. So right here was my professor, Nicola Neretti. I had the honor of meeting him sometime around February, just talking about his science and stuff. This was the lab I worked at. It was in the Center for Biology of Aging. Really new lab at Brown, really fancy equipment. I think there's like a $5 million microscope in there, which they let me use like the last weeks, so that was great. Right here is my mentor, Anthony. He was a PhD student, and he kind of helped me with most of his projects. But all these people are I think they're all postdocs. I think he's the only PhD candidate in there. So very old lab, very smart people. The lab studied cellular senescence. So what was that? That is the opposite of cellular proliferation or cellular division. So it's when cells stop dividing. It can cause a lot of tissue aging diseases, and it can also spread to other cells. But the big implication of it is it's the exact opposite of cancer. So maybe if we understand that, we can understand cancer more. So my first weeks in the lab, they told me I'd be doing a lot of coding. So what was that? I was basically taking images of cells. Like in this one, there's 71 DAPI-stained nuclei. And I was told to write code to analyze it. So I used a deep AI model cell pose, and I outlined each nuclei and printed a picture of it. I was told to do this project on my own. My second week there, my professor said, here's this project, and I was kind of felt screwed. I was texting people in Telluride saying, I don't know how I'm going to do this. But then they actually gave me the great idea of using ChatGPT, and it wrote my whole code, and they thought it was a good idea. They were like, why waste? They said, why waste your time writing code if you can just have a computer do it for you?
As long as they're aware.
It was their suggestion. And then about third week, I got introduced to the big project, which is, say you have a 40 times microscope sample, how do you relate it to an image that's 10 times smaller? So where do these nuclei fit in this? And basic, that. So there's two parts of my work. There was the wet lab work, and there was the dry lab. In the wet lab, I was prepping all my samples for imaging, so I would take liver tissue, cut it up, then I would prep it, wash it, stain it with a DAPI stain or an antibody stain. This process normally took like 2 or 3 days just because it's so time intensive for the chemicals to set in. And then I would mount it on a slide to be imaged. I would take my images, which I would use as data. And then I would write code. So the majority of the time I spent writing code to analyze these images. This was one of my projects where I was setting normalization between 2 different images, the slides I'll show you back. But how can I make these images the same across 4 dimensions, different depths, different spectrosities, all that, and it failed. Science doesn't always work out perfectly, is what my PhD student told me, and he said if you aren't banging your head against the wall for the majority of the time you're coding, you're doing it wrong. So it was supposed to look something like this, but this area is completely wrong, but at least we got that right. And then the coolest part of my lab, I didn't get to this much though, was storm imaging, which I think is stochastic reconstruction optical microscopy. So it would average the photons coming from off a molecule or something when you're imaging it, and you could look down to the molecular level in like a 3D model. So each of these little, each of these little blue stains is actually a nuclei. This kind of shows a little bit better. But I was able to go down to the level like of a single molecule, which is really important for studying senescent protein pathways, especially considering that it's super rare. I think in like liver tissue, it's 4% of senescent cells. So really hard to find these. You need really good imaging when you do find them to understand what's going on. Outside of the lab, I was in Providence, so it was a really beautiful area. I spent time biking down the coast. I found a nice 17-mile bike path. I did a lot of running. This was the sunset about a block from my house, right over the city. It looked like Gotham City one night. I went and checked it out with my roommate. I could get a $10 train ticket to Boston and New York on the weekends, so I did that a few times. And then I'd also go to church every Sunday. There's a beautiful cathedral down in Providence, downtown. which I love checking out. And my roommate was a really cool dude. He was a senior at Brown. Showed me around the city. Took me exploring a lot. Advice: take every opportunity you can, even make ones. Example of this I did during my internship is I started a journal club with one of my PhD students in the lab, Anthony. Every week he'd give me 2 or 3 papers to read about science in the lab, and I'd come back with a presentation I'd present in the lab meeting about what I learned and the figures, and that kind of helped me encourage my knowledge in the lab and made me more useful to everyone else. Refresh your skills and knowledge before you get there. I think most internships use coding, so get better at Python and just refresh it. Ask a lot of questions. I know people in my lab appreciated dumbing down the science to a level that a high schooler can understand because it means they can understand it more themselves. Make an effort to understand what you're working on and take notes every day. And then thank you to everyone here.